Well, I guess it's afternoon now, so I can say good afternoon. My name is Gus Bassani, and I serve as Chief Scientific Officer for PCCA in Houston, Texas. I'm going to start with some comments and then pass it over to AJ, who's got a slide deck to show you guys. First, I just wanted to quickly describe who we are and what we do. In the simplest sense of description, we are a supplier of active pharmaceutical ingredients, excipients, and vehicles, base vehicles for compounding pharmacies, health system pharmacies, and the like. I know there was a lot of questions earlier today about supply chain and the integrity of the supply chain and what are the requirements on APIs used in compounding. In addition to being a supplier, and I'll get into this in just a moment, but over the past 38 years that we've been in existence, we've also been a technical resource for pharmacies that are involved in extemporaneous compounding and personalized prescriptions when those personalized formulations are needed. Kind of a drug. Think of it. We have a drug information center that is focused in on that area. But real quickly on the supply chain aspect of what we do. We have a very in-depth vetting process for raw material and raw ingredient manufacturer selection. Now, we are registered with the FDA as a drug repackager and wholesaler. Section 503A, as Sarah Rothman from FDA mentioned earlier today, requires that active pharmaceutical ingredients used or bulk drug substances used in compounding are made by FDA registered facilities, and that's just part of the. I just want to frame that. That's the baseline. However, there's a number of other things that are important to look at. So when we receive, incoming raw materials, we quarantine every lot and of the chemical that we received, and until the quality control department conducts identification and various other tests specific to that particular chemical, which in our case, we actually do some formulation testing. Again, I'll speak to that in just a moment. So as I mentioned, 503A and 503B require that manufacturers of the bulk drug substances be registered with the agency and in compliance with good CGMP, and also are accompanied by a certificate of analysis. Many of the PCCA's manufacturers that we work with also have drug master files on file with the FDA, which is an extra quality assurance step on our part. We have a series of internal standards that we go by in selecting the manufacturers that we work with, and ideally they would all have DMFs on file with FDA, but that's not always the case. We also have a requirement that an internal specification that the manufacturers have been inspected in the past 3 years, and the reason why that's important is because manufacturers of APIs are only required to register. They're not required to also undergo an inspection before they distribute the product. So it's very important to us that they're not only registered, which is required by law, but also have undergone an inspection, and that is documented. Oh, I just lost my notes here. Here we go. And that inspection also to be inspected by an authorized authority, such as, of course, FDA, Health Canada, European Union, or PICS members. That's the Pharmaceutical Inspection Cooperation Scheme, which was established in 1995 as an extension of the Pharmaceutical Inspection Convention of 1970. EU, each country has their own health authority that inspects to a set of criteria as well. The quality of the chemicals that we distribute is of critical importance for all the reasons that you all have articulated earlier today, and equally critical is how we handle those ingredients. They must be handled and stored properly, and our quality department performs additional testing, including identification after the product has been repackaged. So, as you can imagine, there's a lot of white powders that are floating around, and the wrong thing in the wrong bottle is a big deal, and there's a lot of quality controls in place to ensure that doesn't happen. So on the technical resource side, we have an understanding that of the formulation needs in the marketplace and the aspects of risk that are associated with those formulations that need to be mitigated. Which is why we do have a formulation and R&D team that's focused on addressing the physicochemical stability concerns of these formulations. There's so many variables, especially with topical delivery, that come into play. I think Dr. Murthy did a great job in his presentation articulating all the things that come into play and how a drug is delivered topically. For these pain formulations, we actually have done quite a bit of study on a variety of formulas with regard to the physicochemical aspects of the formulation, of the common formulations utilized in the marketplace, and that's a resource. That's a resource to the pharmacies that are out there. That includes validated stability indicating analytical methods to assess the stability of those formulations when possible, even in bracketed format, so to give some flexibility on the concentrations used in those brackets. We look at content uniformity, homogeneity, and if applicable, USP 51 antimicrobial effectiveness testing to ensure that the preservatives, if it's got a water content to it, that are doing its job. And these are baseline pieces of information. Ultimately, the pharmacies that are making them are responsible for making the preparations and working with their physicians and testing those preparations. But having that good, strong baseline of info is very important. Dr. Murthy spent some time talking about Franz diffusion cell studies, and we've actually performed Franz diffusion cell studies on a number of different preparations in human cadaver skin, in triplicate, and I'm getting close to running out of time. I'll just end with this. And Dr. Day here is going to expand a little bit more on this, but I'm particularly intrigued, and I would point the committee to these studies that were submitted to you on the role of epidermal keratinocytes in the neuromodulation of pain signals. I think this speaks to a baseline viability of a topical approach where you don't actually have to penetrate into and through skin to get a systemic effect. You're actually impacting these epidermal keratinocytes, the nerves, the neuropathic aspects there, and I think that could be an important factor in this whole practice. AJ, you want to take it?